করে দাও।
অডিওটা অফ করছে। তুমি অডিওটা অফ করে দাও।
আমি অডিওটা অফ করে দিলে তোমার সঙ্গে কথা বলবো কি করে?
না তুমি ফোনে কথা বলো না, আমি ফোনে আছি।
আচ্ছা ঠিক আছে, তাহলে আমি অডিও ভিডিও দুটোই অফ করে দিলাম।
Kalpana ji, we can start now.
Yes. Okay. Yes. Good morning, everyone. Welcome to our next seminar for the Science Setu seminar series. Now, this event is being organized as a part of Azadi Ka Amrit Utsav, which is an initiative of the Government of India and 75 years of Indian independence. So these events, since we are celebrating India's glorious history and achievements. With this short introduction, I'd like to request Dr. Acharya to please.
Good morning, everybody. Our today's speaker is Dr. Anulabha Basu. Dr. Anulabha Basu is an associate professor at the National Institute of Biomedical Genomics. Dr. Basu is a statistical and population geneticist trained at the Indian Statistical Institute, Stanford University, and University of California, San Francisco. Dr. Basu's research interest is nested. It has two major components. One component of his research is to understand human genome diversity and evolution with special reference to India. This also provides the framework to look at the multifactorial nature of disease causation, considering host and exposure factors together, and automatically introduces to the second component of his research, which is aimed at identification of genomic regions responsible for differential expression of heritable traits, that is, connecting genotype with phenotype. Dr. Basu's major contribution has been in dissecting human population diversity with special emphasis on India. His research interests include understanding complex genetic traits ranging from position of absolute pitch to cardiovascular diseases and metabolic disorders. His recent interests include artificial intelligence and high throughput sequencing data analysis. So without further ado, I would like to ask Dr. Basu to start his talk.
Thank you so much, Maulinath. Am I audible?
Yes, you are.
Okay. So good morning, everybody. I mean, with that kind of an introduction, I don't think we need to anything else before going directly into this talk. So I'll talk about the reconstruction of our ancestral footprints, which essentially is the evolution of human being, essentially anatomically modern human being over space and time. And the tools that are used for these kinds of inferences, the ones on which I'm going to focus on, are tools from genetics and statistics. So as we all know that reconstruction of human history or human prehistory has been the domain of archaeologists, a little bit about paleontologists, anthropologists, who put together inferences from different kinds of historical and prehistorical sites to come up with a framework of what we knew about prehistory. Molecular biology and genetics has been a relatively recent entrant in terms of understanding of our prehistory. So I'll look at this. This is kind of a reversal of what Dobzhansky has once said, where nothing in biology makes sense except seen in the light of evolution. So nowadays, with molecular biologists and geneticists getting into this domain, what we oftentimes would say, nothing in evolution and prehistory makes sense unless it's looked into the light of biology or genetics. So just to give you a very, very brief introduction, this is a human cell. This is a cartoon from the internet, of course. The human cell looks so slightly considerably different, and it also looks different for different tissues. But this is a cartoon of a human cell with a nucleus here, and within the nucleus there are these chromosomes, which are very, very wonderfully packed inside the nucleus, and the chromosomes contain DNA. There is also a little bit of DNA which is outside, which is in these mitochondrions, and these DNA are the ones where all the information is passed, is inscribed here, and it's also passed on from one generation to another. And we are going to look into these DNA sequences in order to reconstruct individuals' history and prehistory, or a population's history and prehistory. I'll of course focus on human beings, but this can easily be extended to other organisms or other species as well. Because it's humans have 23 pairs of chromosomes. It's pairs because one is inherited from the father, one from the mother. These 22 of them are called autosomal chromosomes, where the father's chromosome and the mother's chromosome generally look alike. There is one pair which is called a sex chromosome, and you must be all knowing that for females, the sex chromosome from the mother and the father looks very similar. For the males, the sex chromosome from the mother and the father looks a little bit different. So members of the same pair of chromosomes are called homologous. Just to give you a sense of this, sense of the packaging. So this has, because it's 23 pairs, which means that there are 46 chromosomes. It has about 0.3 billion base pairs, and each base pair is very, very small. So the cell, which you cannot see with your naked eye, that cell contains this nucleus, which is even smaller than the cell, and within this is this packed chromosomes. And if you take these chromosomes and unpack this and put this in one straight line, that will be somewhere around 6 to 7 feet. So just imagine that our body has so many cells, billions of them, and each of them contains this, within its nucleus, this wonderfully packed chromosomes, which if you extend each of them, they'll be around 6 to 7 feet. So it's a wonderful packaging technology by which it's packed, and not only is it packed for one good way, I mean, you also need to retrieve information for this, which means that you have to access specific regions of the chromosome for each cell, for each tissue, and with respect to certain kind of differences in the environment with which you are responding, to which you are responding. So that that's a slightly different, interesting problem, but for us, the more important for us for this particular talk, what we are more concerned is that DNA sequences of two individuals are about 99.9% identical, and it's only 0.1% which is unique to you and me, which makes you and me so distinct, considering the genetic differences that we have. So again, this is a very simplified cartoon. This is packed in a helical shape, and the nucleotides or the letters in this DNA, or the bases, the nucleotides or bases in the DNA are A, T, G, and C. These are the full names of those compounds, but these are A, T, G, and C. I'll consider A, T, G, and C, and we'll look at this as like a sequence. So this is how essentially an individual's genome would look like. So because it is 99.9% similar to one another, then you can imagine that your father's chromosome and your mother's chromosome are also, they are two unrelated individuals, genetically unrelated individuals typically, and their chromosomes are also 99.9% similar. So if you look at a position on your genome, then in most cases, in most places, they will be exactly the same, because your mother and your father, my mother and my father, has actually contributed the same sequence for both these chromosomes. At certain regions, it will be what your mother and father, what my mother and father has contributed is going to be slightly different, and those are the regions which are called the heterozygous regions, and the other regions are homozygous, which means both the bases are the same. In both the chromosomes, it's only in certain places that these are different, and these differences are different for different sets of individuals. So our genome, this 3 billion bases, of which 0.1, which is also a considerable number of places where we are different. Each of us are different from one another. This genome essentially serves as our data, and we use. So you can imagine that this is, although there is 99.9% similarity, the magnitude of the data which we deal is actually pretty large, and this is where statistics comes into play. That we analyze this data, we identify and decipher patterns from this, and from which we also have theoretical understanding of which pattern means what, and using this we reconstruct what has happened in our past. Just to give you a sense of this, that genomes have different kinds of functions, and the functions can range for I mean, if suppose somebody's chromosome if somebody has an excess of chromosome 21 or a small region of chromosome 21, then it gives rise to certain kinds of disabilities. This is called Down syndrome. If you have a trisomy or 3 instead of 2, if you have 3 counts of a certain region of chromosome 21, then people can have disabilities like Down syndrome. Genomes not necessarily always are responsible for diseases or traits which are very, very important. For instance, if you would like to clasp your hand and put your hand one upon another, you'll see that one of them, putting the right on the left or the left on the right, one of them is more comfortable to you, and this has shown to be something which is genetically determined in individuals. Or a simple thing like whether you have the ability to roll your tongue or not, that is something also which is considerably genetic and hereditary. There are things like earlobes, whether your earlobes are connected to your cheek or whether it's hanging unattached, that depends also on genetics. So what we are going to be more interested in is something which is also called the molecular clock hypothesis. So this was 1963, and Margolish observed that the number of residual differences, this is looking at the protein cytochrome C during that period of time, so it was early on, 1963, between 2 species is conditioned on the time elapsed since the lines of evolution, which means that if there are 2 species which are more closely related, and how at 1963 did we understand closely related? We understood that through mostly by fossil records from morphology, etc. And what we have observed, what Margolish observed is if 2 species are closely related, then the number of differences in the cytochrome C is smaller compared to 2 species which are much distantly related. So just to give you a schematic way in which these are generally the most simplest way in which this is represented is called a tree kind of a representation. So in a tree kind of representation, suppose there are 3 species or 3 individuals here. This is me, this is a chimpanzee, and this is a cow. So what it means here is that in the evolutionary time scale, I'm more closely related to the chimpanzee. And this is where we had a common ancestor, and the chimpanzee and the humans are equally distant from a cow. And the common ancestor to all of us, the cow, the chimpanzee, and the humans, is somewhere much more distant time in the past. So, so if you look at this from a genetic or from a sequence point of view, then it is something like this. That if you look at the genomes of these 3 individuals, suppose a certain region of the genome of these 3 individuals, then what we'll see is you can immediately determine that these 2 are the more closely related species because they have only 1 difference at this region, and this species is much more distantly related to this too. So obviously, if you know this, then this is the DNA from the cow, and these are DNA from the human and the chimpanzee. So this is how it works. The closer related individuals have closely related. The people who have close ancestors in terms of time, their genomes are closer. People who have distant ancestors in terms of time, their genomes are distant. So in going back to this molecular clock, this is again early on. What people have looked at is this is Dayhoff, Margaret Dayhoff. What she looked into is that she compared distances compared to humans. So she took one of the sequence from humans and a homologous region from another species and counted the number of differences, and then there was a function attached to it, and found that and calibrated that with respect to a cow. So a cow and a human, they separated, or the most common ancestor that they had is about 80 million years ago. So that was well known from fossil records. And so she calibrated the differences between human and cow to be somewhere around 80. This is molecular differences. So she took 80 million years ago separation as a gold standard and calibrated this according to this, calibrated that according to this. So if the cow and the humans were separated 80 million years ago, then using fossil divergence time, she would get a baboon and a human human being separation at this. So what she showed and what was shown during that time is that there was a very strong correlation between what the fossil divergence time and the molecular divergence time was. With more technology, with more genomic data coming in, and with more discovery of new fossils, all of these differences that you see here, those differences have also diminished. So this is early on when molecular biologists were entering into the fray, and they were trying to get into understanding of history, prehistory, and using species divergence, evolution using molecular biology and differences. At that point of time, what people had to do is to calibrate all against data from archaeologists. This game has completely turned now. What it has now become is that the inferences are the most robust if you use molecular biology and genetics instead of say anthropology or instead of say archaeology and paleontology. So this is in terms of species evolution, but what I'm going to talk about is something which is much more recent, which is much more data intensive than what was possible for species separation and evolution. It's more about humans. So if you remember the old tree-like structure that we have, and if you have more data, then possibly we can probably expand this to people who are much more closer. So for instance, if you have watched Pother Pachali, you know that these two individuals, they are the brothers and the sisters, they are siblings. So they have a common ancestor only one generation back. So these are human beings who are much more closely related compared to somebody who is a Masai Mara living in the Central Africa, in the African deserts. So using the same logic, if you extend the same logic, what we will expect is that these two individuals who are the brothers and the sisters, they will have genomes or the sequences or the DNA sequences which are much more closer to one another, whereas the Masai Mara individual will have a genome which is much more distant. So can we use this argument in order to use our DNAs, or if you collect samples from different parts of the world, can we use that DNA to rewrite this book of history and prehistory? So this is what was tried, and it is only as recent as 1987 when this was really first tried out in a large scale, and this was tried out in a laboratory in University of California, Berkeley. Rebecca Kahn, Mark Stoneking, and Alan Wilson this was Alan Wilson's laboratory, and these are the authors of this paper. What they did was they collected 147 individuals from 5 geographical populations, and that ranges from Europe, Africa, Asia, Australia, New Guinea, etc. And if you look at this picture to your right, This essentially is a tree. So instead of a tree, what you need to imagine is you need to imagine that this tree is hanging here. So you take this ancestor, you hang your tree here. So these are branches from one arm. These are the extended branches on the other arm. Because it is so extended here, what they have done is they have turned it around and drawn it like this. So this is essentially a tree which starts here. The most recent common ancestor is here, and from here it radiates in different directions. This y-axis or this length is essentially a surrogate of the differences between or difference between this present time to past, as it has been for the other species and the other individuals that we have studied here or that we have discussed here. And what was the surprise here is that if you do this on 147 individuals from many global populations of the world, in these 5 major global populations of this world, what you find is in one arm, you find all Africans. So these are all Africans. And this is taking the mitochondria, which is inherited from a mother to her daughter. To a daughter and son. So mitochondria is inherited from the mother to a daughter and son. And if you look at the common ancestor of these few Africans who might be actually living in small regions which are pretty close to one another, you find that the distance or the common ancestor to all of them, which is this point, is much more, which is farther than all individuals of the world put together and having a common ancestor somewhere here. So the distance of this individual or any of these individuals to the common ancestor is essentially this, which if you look at the sequence divergence is going to be somewhat, and if you translate that into number of years is going to be somewhere at 100,000 at the most. But if you look to this individuals, these are only a fewer number of individuals, and this goes way farther back. So that was very surprising to individuals. That was very surprising to all of them. And what the inferences of this study was is that human beings, or anatomically modern humans, they started their journey in this world somewhere around 200,000 years ago. And that is very likely to be in Africa. So this is quoted from that paper, and this is 200,000 years ago, and this is somewhere in Africa. And not only did they say that this has been in Africa for 200,000 years ago, the first successful movement and first successful colony out of Africa is only as recent as 100,000 years. So depending on whom you talk to, even today, these inferences largely hold very true. The time estimates might vary a little bit, but if you consider the relative time estimates, which is that if we are present in this world for about X amount of X number of years, is as anatomically modern humans are on this earth for about X number of years, then half of the time, the first half is continuous, is almost all completely in Africa. There has been new discoveries, further fossil discoveries, which you might have seen in newspapers very recently, which is trying to push this to 200,000 years to further back, but those things are still preliminary and also needs to be validated further. Now, is this study the one of its kind? It turns out it's not so. So this is 1987. In 1991. From the same laboratory in Berkeley, Alan Wilson's lab in Berkeley, they looked at the hypervariable sequence in mitochondria. And the hypervariable sequence is one region of mitochondria which is slightly variable, which is as the name suggests, is hypervariable. So it accumulates more mutations, quick mutations, which give rise to differences. And with these differences, you get a better resolution to characterize all of this. It also uses a different kind of tree building algorithm. So what they did is that, again, they extended their study. So there were Africans, there were a few African Americans, and then there were non-Africans. So all Africans in this ends with this empty empty white circle, and all non-Africans end with this dark black circles. And what do you see if you construct this tree? Again, this is also a tree which needs to be hanged here. It is also circular because it's just otherwise too long. So the ancestor is somewhere here. And if you look at the first descendants of this ancestor, all these individuals are, as you understand, Africans. So all of them are Africans, and non-Africans are only here. So all non-Africans are eventually related to a common ancestor somewhere here. So this is the common mother of all of these non-Africans, and there are only a few scattered here and there, and everybody else from that on and their descendants are Africans. From here on, descendants are Africans. From here on, the descendants of African. And if you go to this last few branches, the most ancient branches are people who are all from Africa. What is most surprising here? So if you look at this, then all of these individuals, some of them are very closely related, but all of these individuals are from what we call the Bushman peoples or the Kung population. So they have this click kind of a language, and these people are from a very small region in Africa. And in spite of being from a very small region, look at the differences the difference in their ancestry that they have. Some of them are closely related that you are expected to find, and some of these individuals, their ancestors, these are two individuals just living in a village. Just imagine these two individuals, their ancestors go back hundreds. Their common ancestor goes back hundreds of years goes back really essentially thousands of years. So it's close to, so their estimate is around within 166,000 to 249,000 years, and it's about 200,000 years. So these two individuals, suppose from the same locality in Africa, can have an ancestor who goes back to somewhere around 120,000 years or 130,000 years. So this is what people found there, and there is they put forward this theory that people in Africa, they lived, the anatomical modern humans, they started in Africa, they evolved in Africa, they lived in Africa for a very long period of time. In Africa, they did move around a little bit, expanded, contracted, went through different kinds of population scenarios. Then eventually they moved out about 100,000 years ago. And after that, we have colonized the Central World. This is called the out of the Africa theory. It is also called something, it is also called a replacement theory. This is called a replacement theory because it's when you moved out, or when we moved out as anatomically modern humans, the world wasn't a vacuum. It wasn't a vacuum. And there were very, very successful hominid species like the Neanderthals who were living in Europe and in Eastern Asia, which we know from fossil records at around that time. The Neanderthals lived there up until somewhere around 30,000 years ago, as recent as that. And then there were individuals who are other hominids, other Homo erectus species, like some of their skeletons are found. Some of these archaeological records say that they are classified as the Peking man, as a Java man, depending on what the skeletons are found. And they have been very, very successful. They have been living here. Their populations have expanded. They also probably have moved out of Africa at a much distant time in the past. And they were successful in their own ways. They were also, we of course do not know for certain, but they were also very, very well adapted to this environment, because they have been living there for a very long period of time. So it has always remained a surprise to us that if these were so successful individuals, they have large cranials, they're probably stronger than the anatomically modern humans physically, and you do not know whether their brains, their brain sizes were actually not smaller than the anatomically modern humans of that time. And in spite of all of this, what happened essentially is you do not see them. They all got extinct. So So essentially, this out of Africa theory of moving outside of the moving of the anatomically modern humans out of Africa is also known as a replacement theory, because we essentially replaced all of these hominid populations of the Homo erectus populations from all over the world. This is using the hypervariable sequence. In 2000, they did it with the complete mitochondria. The story essentially remains same, that if you take all populations from all across the world, this is the most recent common ancestor to all of them, marked by this arrow and this star. If you look at the Africans, they have much deeper branches, and they are much closer to their common ancestors, to the most recent common ancestor to all of us. So here are the African populations, and this is the common ancestor of the African, the Eve, essentially the mitochondrial Eve, the mother of all mothers. And they have also used an outgroup, which is a chimpanzee outgroup, the closest known ancestor to us, as a different species. And they have at that time used the chimpanzee to kind of hang where this tree is. So it's like hanging that tree. So here is somewhere we have a common ancestor to chimpanzee and humans. So this is the chimpanzee lineage, this is the human lineage, and anatomically modern humans, some origins over here. And as you can see that all non-Africans are here. So it's essentially that people lived in Africa for, what was later shown is, well, so these are the early studies using mitochondria is the one which we discussed. Using the Y chromosome, you can also look at the paternal inheritance, which is from the father to the son, and from the son to the son's son, etc., etc. So if you look at the Y chromosome and reconstruct this tree, you basically reconstruct the same history, which is that we started somewhere in Africa. We lived in Africa for a very long period of time, and about half of the time we are on this earth, and then we moved out of Africa and essentially replaced everything. There is something which is called a haplotype, and there are other kinds of evidence which also started pouring in. Depending on the differences in haplotypes, depending on the similarity, some of these haplotypes are clubbed into what we call is haplogroups. The haplotypes change very quickly, as some of these are defined also on the hypervariable sequences, as we have discussed. The haplogroups do not change that easily. So these are regions where there are certain mutations which are very stable and do not change that easily. Using haplogroups analysis, so these haplogroups are broad classifications. Using haplogroup analysis is the same kind of inferences that we get, which is that it's about 192,000 years before that it all started for anatomically modern humans. This is a line, this is the line separating the tree of Africans and with non-Africans. So all non-Africans are here, all Africans are here. Again, you can see that the length of this tree among non-Africans is about half the time the length of these in Africans. And what they have also seen is that there is these very ancient haplogroups called the L1 haplogroup, which is exclusively found in Africa. And there are ways to date these haplogroups, because as you understand that if you take two randomly chosen L1 haplogroups, the number of differences you see between the haplogroups is going to be very large, which means that two individuals who both have L1 haplogroup are actually very distantly related. So if you take this two individuals, this individual and this individual, then they both have L1 haplogroup and they have very large number of differences between them. So this L1 haplogroup is found only in Africa. The L2 haplogroup, which is relatively much more recent, have evolved on the background of the L1 haplogroup and is also found only in Africa. There's another haplogroup which is called an L3 haplogroup, which evolved somewhere around 100,000 years, a little more than that. And that haplogroup is very closely related to an AIM haplogroup, which has started about 72,000 years ago. The L3 haplogroup is also in Africa, but the AIM, which is a close sister of the L3, is found in many populations all across the world. Similar is the case with an N haplogroup, which is also very close to the L3 haplogroup, which is also found in many populations all across the world. And this extends from populations in Europe, populations in South Asia, populations in Australia, populations in Siberia, to up to all populations which live in Latin America or the North America and the South America, the original Latin Americans or the Native Americans. So what does that signify? What it signifies is that the individuals, so L1 and L2, which are seen in very large numbers in Africa, they really did not move out. So it's only a very small number of individuals who had this L3 haplogroup. They essentially moved out of Africa. And on that background, this N haplogroup and this N haplogroup has evolved. And not only does it says that the L3 is a very small number of individuals moved out of Africa, and this has evolved on that. What it also tells you is after this evolution, after this M and the N haplogroup has evolved, there has been a massive, rapid expansion of populations all across the globe. So the population history essentially remains like this, that we lived in Africa for a very long period of time, and then some people, only a few individuals moved out, and when they moved out, they rapidly expanded in population size. And when they expanded in population size, they also expanded in their horizon. They moved around and colonized different parts of the world. And that ranges from all the way up to the immediate places outside of Africa, the Horn of Africa, and places which we now call the Western Asia, regions where there were the first evidence of settlements, both from archaeology, etc. And from there, we went to different parts of this world. So this individual in this particular study, the individual who's marked in red, this is an Indian individual. If you look at this, there are certain things which are surprising, which is these green individuals are from Australia. As you see that Australians, these are the Aboriginal Australians. They are much more distant in terms of people who are, for instance, these individuals who are from Europe, who are much closer related to one another. So this was something which was surprised that Australia is so much farther from Africa. Compared to that, Europe is so closer. But why is it that all individuals in Europe that they found have an ancestor which is much closer compared to individuals who are far Aboriginal Australians who are so much farther down and so much farther distant from Africa? Why is it that that they have a common ancestor so far back? So now it is with this study and other studies, it has been shown that this is how, if you reconstruct this map, this is how it will look like that about 100,000 years ago. So again, I mean, do not get too much bogged about by the exact numbers, but it's about, some people would now say that this is about something as recent as 70,000. Some people would say that this is more distant, but that's more a technical issue. The more important thing is after anatomically modern humans started, as a species, they evolved in Africa. They stayed in Africa for 100,000 years, or half of the time that they are on this world. Then they start moving out. They moved out in very small numbers. Possibly in very small numbers. At least the successful migrations were out of Africa were in very small numbers, and then they rapidly expanded. The expansion was pretty rapid, that if this is 100,000, then by 60,000 years you find people living in Australia. And there are evidences from archaeology, there are evidence from genetics that people went and colonized Australia very rapidly. We as Indians, we as people living in the Indian subcontinent, we are very interested in this, because if you go from here to here, it's very likely that this early stages or the early waves of migration has been through this land. And if you estimate this is also somewhere around 60,000 years using mitochondria. So there has been the evidence of settlements in Europe is much more recent, which is around 40,000 years. There are many arguments in favor, arguments of why this is so recent, and there are arguments which derive evidence from climate studies, and Europe might have been a little bit more colder, but there has also been there has also been like shifts from ice ages to a much warmer climate multiple times during this. And but anyway, the bottom line remains that the successful migration to Europe has, or the successful colonization of Europe remains as recent as 40,000 years ago. The people's movement, the early Aboriginal people's movement into North and South America has been as recent as 15 to 35,000 years, and this is after the last glacial maximum, which is after the last ice age has started, or ices of the ice age has started melting. This was all glaciers, and people could move this, and that is also corroborated by evidences that this is around that time when the glaciers started melting. And once people have moved in, they very rapidly colonized from here to all the way up to here. All right, so that's a different story, but here is all things put in a nutshell, that the great human exodus started somewhere in sub-Saharan Africa, and the most likely place of migration out of Africa is somewhere called the Horn of Africa, and people lived in Africa for a while, moved out of this and moved to different places, and from there to here. So I use a map here, which is a more common used map here. But if you look at the map we use in trying to look at human migration and evolution, as you can see, that map is a slightly different map where we put Africa in the center of it. So I'm going to skip this for the interest of time here, but there are certain things what we can say in connection to this is that all of this, which we started with mitochondrial data and Y chromosomal data, has now been very strongly substantiated by many other kinds of studies, including the genome-wide autosomal data that we have. And all of this here is one of this. You can see this tree-like structure, and these are all African populations. As you can see, they are closer to the ancestral populations, and these are populations from Western Asia who have substantial African admixture. These are populations from Europe, populations from Central South Asia, populations from East Asia, Papua New Guinea, and these are populations from Native America. So pretty much it correlates with the movement of individuals along these lines, along the lines that were described earlier. Okay, so there are also other kinds of other interesting species evolution which we can see, and this for instance is an obligate pathologic bacteria. This is the tuberculosis, Mycobacterium tuberculosis, which is obligate on human hosts. And if you look at if you globally collect tuberculosis bacteria and sequence them and draw a tree, then the tree looks like this. And if you look at the global mitochondria data and look at this, the tree looks like this. So essentially all Mycobacterium tuberculosis collected from Africa are colored in black here. The branches of this tree, as you can see, these are African individuals for whom you have collected the haplogroup L, which we have already discussed. This is the mitochondria, and these are humans. So this is a tree reconstructed on human mitochondrial data, and this is a tree reconstructed using the Mycobacterium tuberculosis, and there is so much of similarity. So as we moved along, as we moved outside of Africa, we also carried our obligate parasites along with us. Mycobacterium tuberculosis being an example. So what caused this essential replacement? So part of modern humans expanded out of Africa and came out of Africa and populated other parts of the world. They did this. But why would they replace all of them? So is it true that we use the tools more efficiently? Did we kill archaic humans like the Neanderthals, or did we just over-reproduce, or did a combination of all of this? So what is it? What essentially got us to this kind of a success, where there were people who are better adapted, who have been very successful, very widespread, but they just They just essentially vanished, like vanished when we moved out of Africa to their land. Just a clarification that when people moved out of Africa, the world looked a slightly different way than compared to what it looks now, and that is because that a lot of water was trapped as glacial water here. It was ice age and a lot of water was trapped. If a lot of water is trapped, then obviously you understand that the sea level will go lower, which means that what we see in green in this picture is the current map, is close to the current map of the world, but this brown outlines to this green places were also places where people could, which were actually above the sea level. So when people started moving out of Africa, they could actually walk very close to Australia. They could actually start walking, and they can go all the way up to Bali, Sumatra, Indonesia, the tip of Indonesia, and from where essentially Australia is not that far. Not only that, from Australia to many of these Pacific islands, it was essentially, if you could go to Australia, you can also walk along these lines to get to this. Philippines was walkable, so was crossing this. The problem was that there were a lot of glaciers here, but if you could cross this was also land connected, so there was a land bridge. So if you can get to this tip of Siberia, you could actually walk into what is today's Alaska, and then from there onwards. So the map was essentially different. This is how the map would have looked like when people were when the ancient modern humans started moving out. Now this replacement is, so I'll talk a little bit about the Neanderthals and the Neanderthal story and the replacement which has bothered us so much. So when we moved out, as I mentioned, the Neanderthals were they were Homo erectus, and there were evidence of Homo erectus in yellow. And this is the Homo sapiens sapiens, or the anatomically modern humans, moving out in red, and the Neanderthals are in this yellow chrome, and so they were also occupying a very large region. So this is how we moved around, and of course, when we moved around, there are places, there are regions where there is overlap. Now you can only find Neanderthal fossils there, and but the humans are rampaging. So is it true? Is it true that you can only find Neanderthal fossils here? So there were two scientists. The first one we have already discussed, Alan Wilson. Then there is another person who's called Svante Pääbo. Svante Pääbo Alan Wilson has died, as you can see, in 1991. Svante Pääbo is still working, and he's very successful as a scientist. He's the director of the Department of Genetics in Max Planck Institute of Evolutionary Anthropology, and he calls him a Neanderthal man because he had huge interest in Neanderthals. And he has essentially developed a new technology. Again, Alan Wilson also tried this. So what they did was that DNA, which is a molecule, is essentially a very stable molecule. So what they tried to do is that suppose you have a remain, not necessarily a strong fossilized remain, but a remain of something, which is suppose a bone of something which is preserved under relatively good conditions. So this is what Alan Wilson started with a species called a quagga. Quagga is a zebra, is like a zebra and a horse. It's in the horse family. It looks like a zebra, but it's not a zebra. It's an extinct species now. But in the San Diego Zoo of the United States, they had a well-preserved quagga, in a sense that it's not a living quagga, but they have the squelettes and bones of a quagga. The fur and the bones of a quagga. So what Alan Wilson tried to do is, from that bone, can you extract? The question he asked is, can you extract a little bit and have this DNA of the quagga? So they tried to do the sequencing of the DNA of a quagga, which lived thousands of years ago, so 40,000 years ago, 30,000, 20,000 years ago. So this is what he tried, and he came up with a paper in Nature in 1984. Around the same time, in 1985, Svante Pääbo had some material from a fossilized Egyptian mummy, and Egyptian mummies are well preserved. So what his idea was is that he tried to extract DNA from this fossilized Egyptian mummies and would like to sequence it. So how he got access to this is a different story, and we are not going into the details of that. But in 1984 and 1985, there were these two papers which came up, and one claimed that they have sequenced a quagga which lived long back. The other one, Svante Pääbo, who claimed that he has sequenced the Egyptian mummy, which is, of course, is dead and long back. After that, there were many claims, and people went on to their inflated expectation was they could actually, people started claiming that they have sequenced remains of dinosaurs, remains of mosquitoes of that time, and you know some of these in the science fiction. If you have watched Jurassic Park closely enough, the first Jurassic Park closely enough, then there's this, the first part of this is essentially something like this: the DNA of a dinosaur being put into the egg, etc., etc. That's all science fiction, but this is where the inflated expectation around that time was. Turns out that all of this turned out to be false. What turned out to be true was that both Alan Wilson and Svante Pääbo, they both realized that what they claimed as DNA of the mummy and the DNA of the quagga, they were not exactly correct in that. But they continued to work on that lines, and they would like to work on that lines and try to get DNA out of this. And they tried, and Alan Wilson, Svante Pääbo actually worked in Alan Wilson's lab, and they tried together for a long period of time. Eventually, Svante Pääbo came back and set up his own laboratory in Max Planck, East Germany. By that time, Alan Wilson died. But eventually, Svante Pääbo story revolves around Neanderthals. So these are some of the, this is Neander Valley, somewhere in Germany, from where Neanderthals derive its name. And these are places from where there are fossils of Neanderthals, very well known fossils of Neanderthals. This is probably a femur bone from this. There are fossils from the Windia caves. Within these caves, what happens to these, not fossils, these are bones from these caves. What happens in these caves is that the temperature in these caves are relatively constant all around the year, and they have remained constant for a very long period of time. So whatever has gone into the cave, that remains relatively well preserved. It do not get rotten so easily. So it's like a refrigerator, a natural refrigerator in these caves. And from there, he got many of these fossils. This, of course, has to go through a very long and lengthy process by which they get rid of everything else, because there are bacteria working on it. There are other kinds of species which has worked on this, and there are many people who have been on these caves. But they somehow have ways to purify all of this and try and come up with DNA. They were trying to come up with DNA to try and ask the question that what really makes us human, and what is the relationship between Neanderthals and humans. So in that process, this is in 1997. Remember, the earlier one was 1985. In 1997, Svante's group actually came up with Neanderthal DNA sequences, and they did this on the mitochondria, because mitochondria, for each cell there is a large number of mitochondria, so the mitochondrial DNA amount is considerably more compared to the nuclear DNA amount. So they started doing this with mitochondria, and in 1997 they were very confident that they could get some mitochondria out of these ancient genomes. This was the science breakthrough of the year 1997. And what they could do is 123 clones from 13 amplifications. This was a 379 base pair of Neanderthal individual. And they compared that with 478 Africans who live in the world today, 510 Europeans, 494 Asians, and so on. So what they found in the nutshell is something like this, that two anatomically modern humans from any place of the world, if you take them together, they on an average have about 8 differences in these 379 base pairs. So in these 379 base pairs in this mitochondrial region, they have somewhere around 8 differences, okay, roughly on an average, irrespective of where in this world you collect them from. Whereas if you take one individual who's a Neanderthal, or if you take one of these Neanderthal regions and compare that with the anatomically modern human, on an average you observe somewhere around 27 base pair differences. So the number of differences that you observe between an anatomically modern human and a Neanderthal is much more compared to what you observe in with respect to humans, between two anatomically modern humans. So this is what the analysis look like. So you take them in pairs, and you say so these are human pairs. And if you take two individuals, take this 379, compare the number of differences, and see how many of these individuals have them. So 15% of individuals, of all these individuals that they compared, they have somewhere around a difference of 8 base pairs, right? So that's where the mean was. And there are some who have fewer, there are some who have more, but this is the distribution if you compare between a human and a human. Or an anatomically modern human with an anatomically modern human. If you compare a human and a Neanderthal, this is the mean is somewhere around 22, and this is how the distribution look like, the pairwise comparison distribution. If you look at a human and a chimpanzee, then this is somewhere around here. Now, from archaeology and other sources, we know that this was around 5 million years. This difference between human chimpanzee is about 5 million years. So in 5 million years, if you observe somewhere around 55 differences, then then if you calibrate using this, then you can try and come up with also an estimate of this. How many differences would you observe if this is the kind of differences that you see? So it's actually 5.5, somewhere around 5.5 to 6 million is what the estimate is. So you did this, and you say that the anatomically modern humans are very distinct from Neanderthals. The Neanderthals are almost like a different species, and that substantiates our replacement theory, which means that people have moved out of Africa and they have replaced the Neanderthals, who were essentially a different species. So the argument which some people would also give, that humans are descendants from Neanderthals as well, is something which is probably not going to hold true. So they were argued to be a completely different species of hominid species of them. Now the question is that mitochondria you cannot study admixture using mitochondria, but is this possible that anatomically modern humans, they moved out and they had interaction with the Neanderthals, and they could actually mated with them. They could have mated with them and have offsprings who are viable offsprings, and maybe that they passed on their genes from one generation to another. Is this a possibility? So in order to do that, much more recently, there has been some breakthroughs in genetics, in molecular genetic technology, by which much smaller amount of DNA can be used with much more reliability and with much more efficiency to sequence much larger regions of the genome. And in 2009, the intermitochondria of anatomically modern humans and a Neanderthal was sequenced using this technology. In 2010, what people did was having a reference sequence for Neanderthal genome, a draft sequence of Neanderthal genome, which means a complete reference of a Neanderthal genome. As you can see, Sumante Pabo being the lead author, and this was done from one of these bones, from which you can get these sequences out, and they did a complete sequencing of this. So you now have a human sequence. You have many human sequences, and you also have a Neanderthal sequence. And so you can now start asking question: what makes us humans, and did Neanderthals integrate with humans? So what is the evidence of this? So how would you figure out whether they integrate with humans? This is how anatomically the Neanderthal skeleton will look like, and this is what the human skeleton looks like. You can see that there is considerable similarities and considerable differences as well. So if you reconstruct this map, this is where Homo sapiens, and this is the common ancestor of Neanderthals and Homo sapiens. What you find out to our surprise is that more recently, after this 80,000 years, when Homo sapiens and Neanderthals did occupy the same place, there has been certain amount of gene flow from Neanderthals into humans. So, and this is quite substantial, which means that the Neanderthals whose fossils are found all across Europe, if you look at this, you find that the Neanderthal genome now constitutes about 1 to 4% of genomes of all non-Africans, which includes you and me. So the genomes of the Neanderthals are included from 1 to 4% of all non-Africans. So Africans were here. There were possibly no Neanderthals with whom they mated, and they moved out. And whenever they moved out, in this region there were Neanderthals. So whichever direction they went, they probably spent some time here. And when they spent some time here, they mated with Neanderthals and had viable offsprings. And to that extent that they had viable offsprings is that then there was all kinds of admixture events happening with individuals with different kind of Neanderthal and human ancestry mating with one another, and as the Neanderthals got extinct in around 30,000 years ago. But what happened in the meanwhile is every individual who has inhabited for the other parts of the world, they derive about 1 to 4% of their genomes from non-African or from Neanderthals. So our genomes here and mine is 4%, 1 to 4% Neanderthals, and we cannot get rid of it anymore, because as we pass on our genes, we pass on 1 to 4% of Neanderthal genes to our offsprings. So this admixture has essentially blended this Neanderthal genomes within our genomes, and we now have very confident evidence and very confident ways in which we can figure out which region of, suppose, your and my genome is essentially derived from one of these hominid ancestors, and which one is an anatomically model humans. So the out of Africa model is not necessarily a complete replacement theory; it's a leaky replacement. It has replaced the Neanderthals, sure. But in the process, it has been also leaky. So in the process, it has also blended some of this Neanderthal genome within our own genome, or we got it mixed with the Neanderthals as well. So I'll just there are certain kinds of other stories of this, because in the meanwhile, what we also found is that Neanderthals are not the only hominids with whom we had mixed. Using genomic technologies, we have discovered a completely new species called the Denisovans, which was known as the Denisovans because of the place from which the DNA was found, and nobody essentially from archaeological or other evidences or skeletal evidences, it was completely unknown to us that there was a species called Denisovans who are very closely related to Neanderthals, a sister species as we call them, and the humans also distinctly have admixture with Denisovans as well. And what has been the result? Is there any functional consequence of this? There has been some functional consequences, and we are going to discuss only two of them very, very briefly, which is what has happened to this, what has happened because of this admixture. So there are only three groups of individuals who really live in very high altitude, and in high altitude living means you have to live in very hypoxic condition, which means that there is very little oxygen, and you have to use that oxygen very, very efficiently. And people who are able to live in this high altitude have the super athlete genes, and it's called the super athlete gene, and there are populations called the Sherpas. If you are interested in high altitude climbing, then you know that of all people who climb all these difficult peaks, including Everest or all of these high altitude peaks, the best are the Sherpas, and they are the ones who go and climb. And somehow it's unfortunate that they're not even recognized as the climbers. They are recognized as people who just go and fix the ropes, etc., etc. But they essentially can do all the hard work, and many times people are being hosted by those Sherpas, and they really are absolutely fascinating in terms of living in high altitude. So the evidence of real high altitude living is somewhere in Tibet, where much of Tibet is above tree line. It's like a cold desert, and there is very little oxygen there, and you have to really efficiently use this oxygen in order to have a proper physiology to yourself. So they live about 4,000 meters, and it's different from Andean highlands. The Tibetans do not have very high hemoglobin level, which is very strange because hemoglobin, as you know, is the component of the blood which carries oxygen to different parts of your body, different cells and different parts of your body. So anybody who would live in high altitude, typically if I'm taken to high altitude, you will see that my hemoglobin will, in response to this low oxygen, my hemoglobin will raise a little bit. Tibetans do not have very high hemoglobin. They just use the oxygen much more efficiently, and they just use smaller amounts of oxygen efficiently, because if you increase the amount of hemoglobin, then that is not very good, because that makes your blood denser, which increases your chances of getting cardiovascular diseases. So there is a gene called EPAS1, which in the last 3,000 years have been very high frequency and it's found about in 40% in all high altitude Tibetans, and it's one of the fastest genetic swifts that we have observed in humans, which means that this gene probably arose in very small numbers, and in Tibetans or people who lived in Tibetan highlands or lived in high altitude in Tibetan islands, it rose in very high frequency, which means that they are essentially been selected for this gene, or people who had this gene used oxygen much more efficiently, they lived a longer life, they could pass on their genes much more efficiently, and that's the reason the frequency of this gene has increased in Tibetans. Turns out that this gene we have probably inherited from Denisovans about 40,000 years ago. So Denisovans, now there is even more evidence that Denisovans lived in relatively high altitude, and we might have stolen during one of this interbreeding this gene from the Denisovans, and this gene has helped us to live in high altitude in a very efficient way. So this is a study which came up in Nature. There is another story. There are many stories of this kind. There's another story with which I'm going to end this talk. This is the story of the human immune system, and this is the story of the HLA class 1. And if you look at the HLA class 1, there is a certain allele which is found in West Asia, and is oops, sorry. This should not be Denisovans, this should be Neanderthals. So this allele in West Asia was found in a mixture with archaic humans called Neanderthals. The Neanderthals and the Denisovans also have a very similar kind of an allele. So they have been living in Europe and West Asia. So if you think about this, that Africa had a very different kind of an environment. West Asia and Europe had a very different kind of an environment, right? So the pathogens are different. The climate is slightly different. The exposure to other pathogens are different. So the immune system, which is adapted to West Asia and Europe, is not. So okay. So let's put it this way. So the anatomically modern humans who are in Africa and who are moving out of Africa into Europe and West Asia, they had an immune system which is adapted to the pathogens, to the environment of Africa. And when you move out to a different environment, you'd better be equipped with your immune system to be able to fight all of these new pathogens that you encounter during this process. And how have we done this? We have done this a little bit, because otherwise, if you're exposed to a completely new kind of pathogen and if your immune system do not know them, then that might make you sick. That might also kill you. And that is what probably would have happened. So only those specific immune systems which could survive through this and evolve through this would eventually survive. What we did is we did that, of course, but we also took a shortcut in which, of course, not planned, but we got this HLA-B73 allele from Neanderthals, from Denisovans possibly also, and from Neanderthals, and we countered the new environments and the new set of pathogens that we encountered in Europe and West Asia. So So this high frequency allele that we see is essentially also taken from the Neanderthals. So we have been hugely benefited by this. We have been hugely benefited by some of these mixtures which has gone on, because we have sometimes been able to take a shorter route to this, but eventually what has happened is something like this. So this is how the tree looks like. The tree is no longer a tree from one common ancestor to different species. There are intermixing between species, and this is how the tree looks like. That there has been population divergences moving on. So this is populations who live in present day Africa. So this is present time, this is past. And populations recently have gone through expansions, and that's why it has gone through expansions here. This is a time when people have moved out of Africa. These are other hominids who have moved out of Africa. This is a time where Denisovans and Neanderthals separated. They were living as separate species. There has been some admixture between them as well. And then there had been admixture with the Neanderthal branches and the Denisovan branches with anatomically modern humans, mostly during this period, and for the Denisovans also with the Oceanians, which is a very specific and a significantly different admixture that we can see. They around 30,000 years ago, they kind of extinct as species, but we still continue to thrive. And a lot of the reasons why we could thrive is also because of the advantages that we got from them. I would just like to end here with a specific comment here, which is this idea of looking at evolution is essentially a wrong way of looking at evolution. Somebody a few years ago have said evolution is wrong because nobody has seen a monkey to change to a human. Indeed, the monkeys do not change to a human. Monkeys and humans are different species. We had a common ancestor somewhere in past, and from where both of us have derived. It is not a unilateral design, one from a small monkey to a bigger monkey to a human. It's not like any kind of a linearity or a linear progression like this. It is a much more complex scenario. None of these are essentially true. What is true is a much more complicated scenario like this, where the chimpanzees and the hominids, there are many hominids who have separated for a very long period of time. This is about 6-7 million years ago, from which the chimpanzees, the bonobos, have separated with the other hominids. All of these are hominids, and to this end of this tree, for only about the last 200,000 years of this Homo sapiens, evolution do not design us as a successful species. We are successful in certain sense, but we are yet to be proven to be successful in an evolutionary sense, because we have lived in this world for only 200,000 years. There have been hominid species who have lived in this world for a very long period of time, and of course there are species who have been living in this world for a very long period of time. So this is how evolution works. This is how evolution works, where there are common ancestors from which there are separations and from which there are different species arising, and it is not like a monkey starting to walk and getting into humans and all. So thank you so much. Thank you for this opportunity, and I think I'll stop here, and I'll ask Alpana or somebody else to take it on from here. Thank you.
Thank you, Dr. Basu. So there are a couple of questions for you, which the attendees have typed into the chat box. So the first one is by Rachna Acharya, and she wants to know: is the modern human also continuously evolving? If yes, at what is the evidence?
Sorry, Alpana, what's the last part?
If they are continuously evolving, what is the evidence of that evolution, basically?
If they're continuously evolving, what's the evidence?
Yeah.
Depends on how you look at evolution. Yes, they are continuously. If you look at, suppose, simple things like suppose a skeleton of an anatomically modern human who lived in Africa for about 200,000 years, and a skeleton who of a modern human even today, there is certain amount of differences. And not only that, if you just look at the different populations in the world, there are certain differences, which is our response to our different environments, which is our response to different kinds of, which are different adaptations. So these adaptations are in a sense, these adaptations are our responses. These adaptations, at some level, are the molecular responses that are to different environments. So yeah, the evidence of modern humans evolution is a continuous process. It is continuously going on, and if you just look at if you look at it in a small continuous scale, you will see the evidence of adaptation that's happening all the time, all the places. So, for instance, I mean, if you look at suppose simple things like individual skin colors, individuals bone structures, individual structures, etc., etc. Those are adaptive changes that are continuously happening within us. And if you put this into the broad framework, this is a part in which so this is part of the grand synthesis that eventually happens in terms of adaptation to evolution. So
yeah.
Should I
stop sharing? Yeah.
Stop sharing, or should I just keep it in case I need a reference to that?
I think you can stop sharing. That should not be a problem.
Okay.
So there is one more question by Kalol Datta, and he's asking, can you please mention some specific areas where AI or machine learning has been used in solving problems of evolution, and some current research focus in this domain, which can make use of these techniques.
So there has been plenty. I mean, I think the answer to this, if we want to discuss this in a very general forum, this is going to be very, very technical. But there essentially has been many in which people have done this. The basic premise essentially remains oftentimes very similar. The way in which people have used genetics to connect genotype with phenotype, similar kind of premise also remains in terms of connecting history or evolutionary history and dispersals with respect to genomes of individuals. There has been certain application of AI technologies in trying to figure out whether regions in the genome, suppose whether there are regions in the genome of individuals or populations which has been specifically selected at certain periods of time in history. There are certain studies which have used artificial intelligence kind of inferences or machine learning kind of inferences in trying to estimate time of divergence of different sequences. So yeah, I mean, this is all over the place, so to say, right? So if you pick your point of interest, there has been many technologies in which people are using machine learning and artificial intelligence in trying to infer about human history and prehistory. Some of these are going on and done by people whom Kolul might be knowing very well, and particularly the study where regions of natural selection in the human genome is concerned.
Okay. Thank you. There's another question. How Tibetans compensate for decrease in hemoglobin and survive at high altitude? What is the role of PSR1?
Can you? Okay, so is it about the hemoglobin?
Yes.
Can you just repeat the question, Alpana, once again?
Sure. How Tibetans compensate for decrease in hemoglobin and survive at high altitude. What is the role of ESR1?
Oh, okay. So, I honestly speaking, I do not know the physiology of what exactly is the role of ESR1 and why is it that ESR1 is so much more efficient. Not only do I don't know, I think this is not completely understood as far as going to the basics of this is concerned. That why is it that there's certain those particular copies of ESR1 leads to a much more efficient of oxygen usage in terms as far as the cells are concerned. So it's not very well known, not very well understood as of now, but I'm pretty sure that there are experts who can give a much better picture of that compared to what I'm providing here. But yes, I know this for sure that it's not very well understood.
Thank you. At the moment, there are no more questions. We can maybe wait for another minute to see if the attendees want to ask something else. Meanwhile, if for the attendees, if you come up with a question which you would like to ask Dr. Basu, you can mail it to us, and I will try to forward it to him, and I'm sure we'll get back to you and try to solve your queries or address your queries. Don't see any more questions coming up. So I think we can, okay. There's another one by Shubham, and he wants to, can you tell us about COVID Delta variant?
Sorry?
Or the COVID Delta variant?
I don't think, we should discuss this with respect to this topic.
Yeah. I think it's a different thing altogether. Yeah. I think if there are no more questions, we'll close this session here. Thank you, Dr. Basu, for the wonderful talk. And thank you to all the attendees for joining in. We will circulate our next round of information about our next round of seminar series soon. So thank you.
Okay. Thank you so much. Thank you everybody for listening. Thank you for your patient hearing. And thank you, Moli, Arindam, Vikram, Alpana, Priyadarshi. Thank you for making this happen. Thank you so much.
Thank you.
So can I leave?
Yes. Thank you.
All right. Thank you.
Thank you.